para dar inicio a nuestro seminario del día de hoy. Es un placer para mí presentarles a la doctora Laura Botigué, quien nos acompaña hoy. La doctora Botigué es bióloga por la Universidad de Barcelona y obtuvo su doctorado no hace mucho, en el 2012, en la Universidad de Pompeu Fabra, trabajando sobre la historia de las poblaciones humanas en el norte de África, bajo la supervisión del profesor David Comas. Este trabajo le permitió caracterizar la arquitectura genética de las poblaciones del norte de África, y después se incorporó a la Universidad de Stony Brooks, ya como postdoctorado en el laboratorio de la profesora Brenna Hannan, becada por el gobierno de Cataluña, donde continuó su investigación en poblaciones humanas, pero donde también inició una línea de investigación sobre DNA antiguo en perros, que nos platicó ayer una historia muy interesante, con el profesor Krishna Verama. Y después de su postdoctorado continuó trabajando con DNA antiguo, pero enfocada más en plantas y el proceso de domesticación. En el 2017 inició una colaboración con investigadores del College of London, ya trabajando con trigo antiguo, y se incorporó al Jardín Royal Botánico en el Reino Unido. Un año después se unió ya al Centro de Investigaciones en Genómica Agrícola como Junior Group Leader, y en el 2020 obtuvo una beca muy prestigiosa, que es la Ramón y Cajal, que le permitió considerar su grupo de investigación en cultivos antiguos y domesticación. El año pasado obtuvo la titularidad como científica del Consejo Superior de Investigaciones Científicas de España, y bueno, su grupo claramente se dedica actualmente ya a la genómica de cultivos antiguos y la domesticación. Estudia la evolución del genoma de los cultivos para comprender los cambios que experimentan las especies silvestres al adaptarse a la agricultura, y para ello aplica evidentemente teorías de la genética de poblaciones y bioinformática y el análisis de variabilidad genética. Sus aportaciones al campo han sido muy importantes y se han publicado en revistas como PNAS, PLOS Genetics, Nature Plants, entre otras. Y bueno, me da muchísimo gusto que haya aceptado la investigación y que estés acá. Muchas gracias, Laura.
Que me han tratado muy bien, y en general también la curiosidad de los estudiantes, ¿no? En la clase que estuve dando ayer, en general, ¿no? Muchas preguntas, muy buenas preguntas, y esta curiosidad genuina, que es como una brisa de aire fresco, ¿no? para la investigación y la ciencia, así que enhorabuena por el trabajo que estáis haciendo. Voy a empezar la charla, and I'm going to switch to English, that I feel a bit more comfortable with it. First of all, I want to talk about wheat and the importance of wheat. It's clear that wheat has a very high economic importance. Maybe not so much here in the Americas, where maize is much more consumed than wheat, but especially in Europe, because its origin was in the Fertile Crescent. It's a very heavily consumed crop. It's a staple crop. And also in terms of calorie intake and also the protein intake from cereals, it stands out as one of the most important cereals. But also it has a very a high historical importance. So wheat was among the first crops that was domesticated. It was domesticated in the Fertile Crescent. And with it, the whole process of the origins of agriculture started. And then its expansion, which was a very important process for human civilization, human culture, and the societies as we know them today. Without agriculture, we would not be the way we are. No, in a summary, in a short summary. Wheat is also important, or it's nice, because its wild ancestor is only able to grow in the Fertile Crescent. It has a very restricted geographic area. And then even when it was domesticated and it spread with agriculture, its wild ancestor stayed there. It did not spread forward. So in this sense, from an evolutionary point of view, if you think about it, you have wild wheat growing there, and then you start to have agriculture that spreads everywhere in the world, And nowadays, from this same genome that is only able to grow in there, you have wheat growing in Central Europe, you have wheat growing in Ethiopia, in India, and in so many different agroecological systems. And therefore, the genetic basis of these adaptations must be in the genome, right? So in this sense, that's also why I'm interested in understanding wheat domestication, but also its history and its evolution throughout the process of expansion. So a very brief history on wheat evolution. There are actually two different kinds of wheat that were domesticated in the Fertile Crescent, but I'm just going to talk about the one that is more famous today. So it starts with wild emmer wheat. This is a tetraploid, so it has an A subgenome and a B subgenome, and it has two characteristics. So the first one is that it's brittle rachis. So that means that it has its natural and dispersal mechanism of seeds. This means that when the grains are mature, they just fall, they grow, they fall into the ground, and then they grow next years or in 2 years or whatever. And it's also hulled, that this is another trait that if I have time, I'm gonna talk about it a little bit later. Then wild emmer wheat was domesticated and appeared the domestic emmer wheat, also a tetraploid. And there are more differences, but the main difference is that this natural seed dispersal mechanism is lost. So this one has a tough rachis, it's not brittle anymore. And we know that now that this trait is determined mostly by two genes, which are the triticum brittle rachis. And it's actually like the same gene, but it's the copy that exists in the A subgenome and the copy that exists in the B subgenome. And this is one of the most diagnostic domestication traits that is used to determine if wheat is wild or it is domestic. So this went on. This domestic wheat was cultivated for thousands of years in Europe. It also spread into Africa, into Asia, particularly like Central Asia and towards India, arriving into China as well. No, sorry, not emmer wheat. It did not arrive into China. And then at some point, this emmer wheat is transformed or evolves into the durum wheat. It's a free threshing wheat. It evolved into one of the ancestors of durum wheat, which is the wheat that we now use for pasta, for instance, or couscous. This is durum wheat. It's also a tetraploid. Of course, it has this domestication trait, the brittle rachis, but it's also free threshing. It's one of the characteristics that differentiate this ancestral wheat from the modern one. Then also the durum wheat hybridized with Egilopstowski, which is called also goat grass. It's a wild grass that exists around the Caucasus, but also in some parts of Southwest Asia and Central Asia. And then it hybridized, giving rise to bread wheat, the hexaploid, and the wheat that is probably the one that is mostly grown nowadays. So just for clarification, for this talk, I'm going to focus on the ancestral wheats, so the wild emmer wheat, and the first wheat that was domesticated, which is the domestic emmer wheat, and that they are both tetraploids. So if we look first into the genetic structure of these wheats, what we see is that, as I said, the wild wheat grows only in the Fertile Crescent, and it's divided into two main populations or subpopulations, which are the southern Levant and the northern Levant, which are zoomed down here. The southern Levant is more the area of nowadays Israel along the coast, and then the northern Levant reaches Iraq. There are some samples from Iran as well. It goes into Turkey and Syria and Lebanon. And then we have different traditional land races of emmer wheat that we were able to obtain from like seed banks and germplasm collections. And there is a Mediterranean population, a population from Eastern Europe, the Caucasus, and then this group here that I'm going to talk more later that is they called initially Indian Ocean group. And if we are going to put this genetic variability in a tree, what we see is that the samples, the wild samples from the southern Levant are very diverse between them. There is a lot of diversity in this pool of wild populations. The ones in the northern Levant are less diverse between them, and they are the ones that are closest to all the domestics. So in 2018, we started this work, 17-18, We obtained an ancient sample from Egypt, it was a 3,000-year-old wheat. It was in a museum, in the British Museum. And what we wanted to see is whether this ancient wheat looks similar to these modern land races that we had today. So when we placed this ancient emmer wheat into the phylogenetic tree, what we saw is that it was very close to this group that was called the Indian Ocean group that had only 4 representatives. So it was very limited compared to the rest of the domestic accessions. And there were some samples from India, some from Turkey, and some from Oman. And it kind of made sense because Egypt is towards the south, but it was surprising also that we detected an event of gene flow between some wild wheat from the southern Levant and this ancient sample. So that was intriguing. It could mean that there had been like a different history or a different evolutionary history between these ancient wheats and the modern wheats that we see today. And also something surprising to us was that this wild emmer wheat from the northern Levant is actually closer to these domestic wheats than this Indian Ocean group. So the Indian Ocean group appears as an outlier when you compare all this clade here. And this was highly surprising because our idea was that the domestics would all tend to go together. So something else that we wanted to check, going to this domestication gene, the triticale brittle, the brittle rachis, is whether we could see the signal of selection around these genes that they had been characterized. So what we did was we took windows of the genome around around the 2 genes. And then for each window, we calculated the average minor allele frequency that there were there on the SNPs, with the idea that if you have had a selective sweep, you will have a lot of fixed SNPs around the gene, and therefore you will have only alternate alleles if you want. And this is exactly what we saw. So in black, you have the minor allele frequency of the ancient wheat, on orange, the one from the domestics. And you can see here that around the gene, there is like a huge region where almost all the frequencies, all the SNPs are fixed, with some exceptions in a few of the accessions. Whereas in the wild, you see much more variation and neutrality, meaning that there are like very different frequencies on those mutations around there. And this is very, very evident in the B subgenome. And it's a bit less evident, but still you have here quite a lot of loss of diversity in the A subgenome, meaning that even after 10,000 years that these genes were selected, we can still see the footprint of selection. Then something else, continuing trying to get this question of, is this ancient wheat really close to the modern ones or not? What we did was apply several selection statistics. So these are 3 quite common selection statistics that are FST based on genetic differentiation. This is P versus Watterson theta, which is also, these are selection statistics that are based on the variability and on the way that you observe variability. And then if you choose the outliers of these statistics, you have candidate regions that are under selection. So in the end, what we have here are windows that are putatively under selection in the domestics. And what we wanted to see is whether the ancient wheat behaves in the same way as the domestics do. And this is what we see. So we have here a high concordance for all the putative regions that are under selection compared to the wild. So this means that the ancient wheat had undergone the same selective pressure than the domestic wheats that we see today. And then the next question was, okay, what about the neutral regions? If they have undergone the same selective pressure, this means that probably they share the same history. They suffered the same evolutionary fate. Therefore, in the neutral regions, you would expect the same pattern. And this is not what we see. So when we look into the neutral regions, we see that the concordance, except for some outliers, the concordance is more or less the same as the wild. And at the time, this meant that most likely this Egyptian wheat had a rather different trajectory or demographic history than the modern wheats. So then, since we had observed this, and we also had observed that this Indian Ocean group behaved like a little bit weird, that we were not able to explain why it behaved why it was so different from the other domestics. We decided to look into this. So we used a data set that had been published in 2020, where there were 57 wild and domestic MR accessions. And the great advantage was that there was a huge overrepresentation of the African continent. There were more samples from Oman. And this was very good for us because this was highly underrepresented. Until now, all the MR with variation had been based mostly on land races from Europe. So when we analyzed this data, we confirmed that this Indian Ocean group was weird. So this is a principal component analysis. And we see, as expected, that the wild wheat from the southern Levant is more differentiated than the others. But to our surprise, when we looked into the second principal component, this wheat from the Indian Ocean group that also now included Ethiopia was the one that was more differentiated from the domestic wheat from Europe, but also from the wild wheat from the northern Levant. Another evidence of this is that when we applied that mixture, which is a software that assigns ancestries to the different wheat accessions, grouped the wild samples from the northern Levant and the domestic samples from Europe together. And the Indian Ocean samples, they were apart. They had their own ancestry. So since we had detected gene flow, but at the time, in the other study, but there was very little sample size, we said, okay, let's try and model now gene flow again, now that we have like a larger data set. So we first did a phylogenetic tree to see where the samples, how the samples were placed. But then we also applied this model that it's called tree mix that builds a phylogenetic tree on these groups that appeared in the phylogenetic tree. And it allowed edges of migration. So it allows for migration events. And you can model different migration events and then check how good is the model compared to the data. And the results that we got is that the best model was a migration event from the wild southern Levant into this entire group of Indian Ocean wheats, that at this point we just stopped calling them Indian Ocean and we call them Southeast, because in the end, it's one of the dispersal routes of wheat after its domestication into the south of Africa, then the Arabian Peninsula, and then towards India. So from now on, I will call them Southeast. Then another thing that we wanted to look into is, okay, we have broadly two different domestic germplasms, the one that is from the southeast and the one that is from the northwest, which is mostly Europe. And we have two different wild populations. So let's try to model these wild populations so that they will give us the domestics that we observe today. So what proportions of each wild population we need to create each of the domestics. And what we saw is that for the domestic northwest, from the accessions that are broadly from Europe and the Caucasus, we had around 70% of the ancestry that belonged to the wild northern Levant, and between 20% on average of ancestry from the southern Levant. But when we went to model the domestic southeast population, we had around between 40 and 50%, or 50, 60, of the genome was from the wild northern Levant, and we had like an increase of southern Levant contribution. So we were confirming this gene flow from the southern Levant into the domestic southeast, but we were also able to determine what proportion. So on average, it had 20% more wild southern Levant ancestry than the other domestic. With this software, we were also able to give an estimated date of hybridization. So at what time did these two wild populations merge to make this domestic population? And from the northwest, the estimated domestication time was around almost 10,000 years ago, which matches very, very well with the archaeological record when the first domestics are observed. And for the domestic southeast, it matches 6,000, well, it gives us 6,000 years ago, which is just before we find the first domestics into Egypt. So this matches with the south route of dispersal of domestic emerald. Then we wanted to continue with this idea, and we said, okay, the domestic southeast has this extra contribution of wild emmer from the southern Levant. Is this adaptive? Is there a selective signal? Because we observe this today, like we can observe it today in the modern wheats. So did this have an adaptive effect? So what we did was select all those regions in the genome. So we use this DXY statistic, which is also a statistic for genetic differentiation. And we selected all the regions of the genome for which the domestic southeast is very close to the wild southern Levant, and it's very different from the domestic northwest. So it's uniquely close to the wild southern Levant. So this gave us 247 windows, and then we looked at the genetic variability in these windows, and we found 300 SNPs that had a predicted high or moderate impact in the biological function. So these are mutations that could have a high effect on the biological function. We ran another test statistic to try to have like a different approach. And this is a cross-population and selective statistic. And the idea is that we want to look at patterns of genetic variability that match selection and that they are different and between the two domestic populations. And what we saw is that it stood out, this window that is found in chromosome 4b. So this is a 200 megabase window. So just a reminder here, wheat has chromosomes that are 800 megabases long. And then as a parenthesis, everything is a pain. But this also implies that as you move away from the edges of the chromosomes, the recombination rate is very low. Wheat is also a selfer, and that means that the recombination rate is even lower. And therefore, there are little chances for it to, create variability. And probably that's why we have this huge region of 200 megabases for which we have 71 genes that are affected by this high or moderate effect SNPs. Then what we did was run a gene ontology analysis for these genes. And what we got is this classification here that it's organic substance and chloroplast metabolic processes. And then we were looking into it in the bibliography, and we saw that the chloroplast actually plays a very important role in abiotic stress, and particularly to exposure to light. So this is kind of a cool result that we have no functional validation for it, but it's a nice candidate to, dig deeper into it, because of course, if wheat has to move into Egypt and Ethiopia, you would expect that you need resistance to high light exposure. Another thing that we did, we have collected more ancient samples from Egypt, and we have done exome sequencing data on them. And what we wanted to do, we just did this as a sort of validation because we wanted to make sure that the signal that we were seeing is actually also ancient and not something that occurred like recently. And we looked into this region in chromosome 4b, and we built a haplotype network with the variability that we observed. Of course, it's ancient data and we have a lot of missing data, but we can still see the signal that this is the ancient sample, and it's way closer to the domestic emerald wheat from the southeast and to the wild wheat from the southern Levant compared to the one from the north, both, the wild and the domestic. So this signal was already present 2,000 years ago. So just to wrap up in this part, we have now built a sort of model of domestication of wheat, incorporating also information that we know from the archaeological record, which is very rich. And it is that wheat, so you need, I talked about the brittle rachis gene. You need, this gene has two copies, one in the A subgenome and the other in the B subgenome. You need to create loss of function mutations in these two genes independently to get the domestic phenotype. So with all the work, state-of-the-art work that there is, and the work that we have done, The model of domestication is that one of these copies of this gene, one of these loss of function mutations, appeared first in the southern Levant. You start to have proto-domestic wheats, and that means wheats that show an intermediate phenotype. Then after some time, the mutation, the loss of function mutation appears in the north, and then you also have an intermediate phenotype in the north. Then these wheats from the south travel into the north, they hybridize, you get the full domestic phenotype, and then from that point on, everything goes like rather quickly. Once you have the fully domestic phenotype, it moves into Europe and creates these domestic Northwest land races that we see today. This matches the fact that domestic Northwest is closer to the wild wheat from the northern Levant. Then this full domestic also goes south. Once it goes in here into the south, it hybridizes with the proto-domestics that there were there, acquiring this extra component of wild southern Levant ancestry. And then it moves into Egypt, Arabian Peninsula, and India, creating the domestic Southeast land races that we observe today. So this is a bit the history from the first part. I think that I'm good with time. Yeah, I'm gonna talk briefly about this second project. It's very brief because this is something that we are working on now, but I like it because it's about the free threshing trait, which is another trait of agronomic interest in wheat, and it has to do with the processing of the wheat. So the first domestic wheat, if you remember, the emmer wheat is a hulled wheat or gloom wheat, and this means that once the spikes get released, they actually there are these like leafy structures. The grain is inside here, but there is all this structure that you need to decompose to free the grain. And this is very difficult. It's really annoying. You have to really be very strong to actually get the grain out of it. And it's very time consuming. In opposition, you have the free threshing wheat, which has a characteristic in which once the grain is mature, since it's domestic, it will remain attached in here, in the rachis. But if you just batter it, it kind of decomposes, everything falls apart. And then it's very easy to free the grain from the chaff. So this is the free threshing trait. There are many studies that have been studying this trait. And 3 QTLs have been associated with this trait. One is in chromosome 5A. This is the apetala-O2 gene that has been already described in cereals. They call it the domestication gene, actually. And it has like a lot of pleiotropic effects. And then there are 2 peaks in chromosome 2A and chromosome 2B. And we wanted to see whether we could find the causal mutation of this. Like it was a very naive idea at the beginning, but we said, okay, we have a lot of genomic data. Why don't we try to just find the gene that is causing this mutation? Also because it's kind of nice because maybe they are the same gene. It's like the brittle rachis for the location. It could be the same gene in copy subgenome A and subgenome B. And we started with subgenome B, and we found like a huge inversion in that co-localizes with the trait, which is very exciting because then it's like, oh, the inversion probably is actually causing the phenotype. We have validated this. So this is 2 genome assemblies, one free threshing and one hulled. And we are just doing an alignment of the regions. And there is kind of continuity until you get into the inversion, and then it continues. So we have done this. We have a limitation that we have only one proper genome assembly of a hulled wheat. And it's not very good. It has it was based on short-reach sequencing data, and it's poor quality. And this is some 10x draft genomes that we created just to validate the inversion because we didn't know whether it was something unique to the reference genome or it was real. So the inversion is real. It has around 6 megabases. It's around 6 megabases per long. And there are around 55 genes inside the inversion, and 2 of them are located near the boundaries. And now our job is trying to figure out which of the genes, of these genes, can be candidate of causing the phenotype. So one of the first things that we did was just create this sort of synteny analysis between the best free threshing genome that we have that was recently published. It's a telomere to telomere genome assembly. It has no gaps. And our not so good whole genome. So you can see here that we have a lot of ends. So regions that we don't know what's going on inside, but you still can see the signal of the inversion, right? So this area here maps towards the end here, and this area here maps in here. So we looked into the abundance of repetitive elements to see whether they have played a role in the reordering of genes. We have also looked at where are genes located in these genomes. And we are in the process of doing so. So we have the genes here. We are in the process of annotating the repetitive elements on the halt width, and also seeing how close they are to genes to see if they could affect the function. And this is like on one hand, we are doing this like genomic analysis. We are also going to do RNA-seq analysis to see whether we see differential gene expression during spikelet development between the two kinds of width. And with the hope that in this sense, it's easy because we already know what are the genes nearby the inversion. So we just need to check whether these genes specifically show differential gene expression. And then something else, a bit of more population genetic analysis. We wanted to see whether the inversion showed evidence of selection or not, like was it adaptive or not. And for this, we did a haplotype network for the SNPs that were upstream of the inversion, and a haplotype network of the SNPs that are downstream of the inversion. And what we see, And what we see is that these are all the free threshing widths that cluster together. And then the hold ones are more like spread in the network and do not show that much clustering. So in particular, like these haplotypes here, this means these big circles mean that there are a lot of accessions that share the same haplotype. And for the downstream, we see more or less the same, a lot of clustering of the free threshings. And then the ones that are hold that are more spread. So also the yellow ones are also hold widths. So you can see like the clear difference. So this clustering of the haplotypes is compatible with like a rapid expansion of or a loss of variability of this inversion. And this also implies that it had an expansion, and therefore most likely it was selected. And this is my last slide. I want to thank you all for your attention. The people in my group, Alice and Costas, are the ones that worked. Well, Costas is still working very hard on the inversion. Alice was the one that did the work with the MRWIT, and also all the collaborators. And I will be happy to take questions.
¿Preguntas?
Lo voy a hacer en español.
Perfecto.
Seguramente me voy a equivocar de cuál es cuál, pero me llamó muchísimo la atención. Será porque trabajo con cloroplastos, pero me llamó muchísimo la atención esta parte que dices de la especiación, que, bueno, de estas características que parecen todas ir a algún aspecto de
adaptativo
de plasma.
Ajá.
Y mi pregunta nada más es, en tu análisis no pudieron ver algún gen en particular que fuera el que, porque muchas de estas cosas van a ser secundarias, que dan como resultado pues adaptación fotosintética, luz, lo que sea, pero pudieron ver algún gen en particular en el que
sí, mi limitación es que yo de cloroplastos no sé nada, o muy poco. Entonces, si hubiera genes que se sabe que están implicados en este metabolismo, yo a priori no los conozco y no sé cómo se llaman en trigo, que esto es otra historia, porque en realidad el proceso, bueno, primero voy un poco atrás, que la aproximación ha sido, nosotros vemos esta región que está bajo selección y entonces una opción es ir gen por gen y mirar por qué nos da una señal selectiva. pero como primer pase lo que hicimos fue ver si hay un enriquecimiento de alguna vía metabólica, ¿no?, en esta región que explique esto, ¿no? Y esto es lo que nos ha dado con el Gene Ontology. Después lo que hicimos fue ir a ver estos genes, cómo se llaman en Arabidopsis y en qué procesos están implicados, pero no pudimos ver nada que dijéramos fue este o algo así. Pero bueno, si supiera cómo se llama lo que estoy buscando, ¿no?, entonces igual sería más fácil también.
Sí, es muy interesante lo que estás viendo, porque finalmente, o sea, van hacia, si entiendo bien, son aquellos que migraron a África.
Correcto, correcto.
Entonces, la cantidad de luz pues va a ser mayor, pero también
Exacto.
la temperatura. Y por eso te preguntaba yo,
Claro.
adaptaciones de que va a haber pues varias cosas que me parecen súper
Claro.
interesantes.
Claro, a mí me alegra porque al final uno usa el Gene Ontology para ver qué sale, ¿no? Y a veces salen cosas que tienen sentido y a veces cosas que no. Y nosotros a priori cuando vimos lo del cloroplasto nos quedamos un poco así, pero después viendo la literatura bibliográfica sí que vimos que tiene un rol importante. Y entonces, ¿no?, sí que estamos cautelosamente emocionados porque igual sí que es verdad que ha sido adaptativo y podemos encontrar exactamente qué es, ¿no?, de esta región, qué es lo que lo hace adaptativo. Pero necesitamos, o sea, podemos hablar después porque necesito más conocimientos moleculares para dirigir la pregunta, ¿no?
Oye, y mi segunda pregunta es, o sea, es claro que esta mutación en el raquis para que se mantenga y no se caiga es fundamental y no nada más ha sido tal vez para trigo, sino para muchas especies de domesticación.
Sí.
Pero aparte del raquis, ¿hay alguna otra, algún otro adaptación? No, pues,
sí, se sabe. Sí. Está una que es también muy evidente, es el tamaño del grano, el tamaño de la semilla, y de hecho hay no sé, creo que el gen concreto no se sabe, pero sí que hay QTLs que se sabe dónde están, que están asociadas al grain size, y también hicimos el mismo análisis, pero bueno, que no lo he mostrado. Pero después también hay la inhibición de la vernalización para asegurarte pues, que cada vez que germinas el grano te crezca el trigo. Y la sincronización de la floración también es muy importante para que todo el mundo te madure a la vez y no tengas un campo de trigo, ¿no?, con distintos timings.
Mi pregunta sería, o sea, tú demostraste de una manera súper bonita cómo las dos mutaciones en el raquis, dos mutaciones independientes, estuvieron ancestralmente a esta diversión de los semidomesticados. Y en todas estas...
Esto no lo he hecho yo, ¿eh? Este mérito no me lo puedo atribuir porque ha sido un trabajo súper bonito que han hecho unos investigadores de la Universidad de Haifa que han hecho esto. Una vez caracterizado, ellos publicaron el genoma de referencia del trigo salvaje y hicieron un ejercicio para encontrar estos genes que sí que se habían caracterizado en cebada, y entonces buscando homólogos de los genes de cebada que ya se conocían, encontraron estos del trigo. Y después hicieron otro estudio, que esto en la clase lo comentamos ayer, que es un estudio que mira toda la variabilidad genética alrededor de este gen, y lo que quieren hacer es, ellos ven las mutaciones que existen en los domésticos y van a encontrar el trigo ancestral que tiene todas las mutaciones de los domésticos, excepto la que causa el fenotipo, de modo que es el ancestro directo. Y bueno, al final sí, encontraron los trigos para los dos y a partir de aquí se pudo construir esta teoría. Sí.
Gracias.
Muchas gracias.
Eso es muy interesante. Siendo evolucionando un poliploide, hay un proceso de selección para seleccionar eventos como inversión más que como mutación para justamente divergir los diferentes homeólogos.
Esto se está mirando ahora porque la caracterización de inversiones o variantes estructurales y todo esto solo ha sido posible desde que la técnica del long read data es affordable, ¿no? Hasta ahora era una limitación muy importante. De hecho, no se sabía que había una inversión hasta que no pudimos mirarlo. Y de hecho, cuando descubrí la inversión, no pude continuar con el proyecto porque solo tenía short read data. Sí que hay estudios que han estado mirando, tipo que han construido el pangenoma, ¿no? de distintas variedades de trigo, y era bread wheat en realidad, y sí que han podido identificar cómo hay algunas inversiones que además parece ser que sean como introgresiones de otras especies de Triticum, pero que no se sabe muy bien si son ancestrales o han sido eventos domesticados, pero sí que es verdad, no sé si es por el hecho de ser poliploide o no, pero que la variabilidad estructural es muy importante para los caracteres en general, para los fenotipos, para la variabilidad fenotípica, es algo que cada vez tiene más fuerza.
Voy a leer una de YouTube.
¿Vale?
La doctora Alejandra Alicia Covarrubias pregunta cómo se compara la historia evolutiva del trigo Emmer contra otros tipos de trigo.
No puedo responder a esta pregunta porque el otro tipo de trigo es el einkorn wheat, que es un diploide. Y bueno, a nivel genómico sé poco de su domesticación. A nivel arqueobotánico, creo que se domesticó más o menos, no, esto sí que lo sé, que se domesticó más o menos, o aparecen las variantes domésticas, aparecen más o menos al mismo tiempo, o sea que fue de modo simultáneo, y más en general, hace unos 10.000 años aparece el trigo, que es uno de los primeros domésticos, pero de modo simultáneo está la cebada y está este otro trigo, que es el einkorn wheat, pero que no tuvo tanto éxito. Y aquí no sé si esto es 100% verdad o no, pero creo recordar que leí que el einkorn wheat está más adaptado a climas más fríos y tuvo más éxito como hacia el Cáucaso y estas regiones más del norte de Europa, pero que al final el emmer wheat acabó siendo más un staple crop. Vale. Mira.
Hola Laura, mil gracias por tu plática. Siempre me encanta escucharte y siempre cosas nuevas y súper interesantes. Y un poco yendo a tu último resultado sobre la inversión, bueno, a sabiendas que pues el ADN antiguo y la paleogenómica tenemos la principal limitante de las lecturas súper cortas, pero ¿crees que sería posible con suficiente secuenciación y obviamente encontrando aquella muestra de la temporalidad precisa, pero poder determinar el momento en el que en la historia evolutiva ocurrió esta inversión? ¿La estás buscando?
Claro, es que bueno, tú ya sabes que la mayoría del trigo está carbonizado y que esto es el mayor factor limitante. Yo creo que un approach posible podría ser si se consiguen encontrar variantes diagnósticas, ¿no? Es como el GWAS, variantes o marcadores de QTLs. Usar SNPs que sean diagnósticos de la inversión. Y entonces utilizar estos SNPs como un proxy de si yo observo estos SNPs en este trigo, probablemente estará la inversión al lado. Esto podría ser un approach, pero primero habría que tener una caracterización súper buena de la inversión y de la variabilidad que hay en muchísimas accessions de trigo. Pero aún así, yo creo que puede ser posible intentar modelar la fecha de la inversión, que siempre es un approach más bien informático, pero cuando hay la inversión, durante un periodo hay una caída de recombinación también dentro de la inversión, ¿no? Porque no pueden recombinar. Y creo que esto sobre todo en los breakpoints. Y si consigues modelar esto bien y observar la variabilidad genética allí, en teoría, estas son muchas teorías, creo que podrías llegar a modelar o estimar más o menos cuándo ocurrió, que sería un resultado súper bueno.
Bueno, yo tengo una pregunta, muchas gracias por su plática, que no está relacionada directamente, pero en base de datos genómicos o no genómicos, si existe alguna información sobre tendencias durante domesticación en cambio de sistemas radicales en trigo. Por ejemplo, si puede inferir en base de los genes involucrados en civilización de las cosas que podrían ser útil para entender esto.
O sea, la evolución del sistema radical en trigo durante la domesticación y la expansión. Yo creo que no, o al menos no me viene a la cabeza ahora que haya como bases de datos, pero si se conocen bases genéticas, ¿no?, que estén asociadas a cambios en el sistema radical, entonces sí que se podría estudiar la variabilidad en estos genes a lo largo del tiempo, o sea, hacer como una aproximación inversa.
Simplemente en base de variedades o de razas que estaban.
Ajá.
¿Y existe alguna información similar en cualquier otra especie?
Sobre el sistema radical.
Sobre durante todo este caso.
Sé que hay gente, bueno, hay investigadores que trabajan con Brachypodium, que es salvaje, pero sí que han estudiado evolución o diversidad de sistema radical en Brachypodium, que podrían servir como modelo para después estudiar la domesticación. Pero no digo que no exista, digo que no lo conozco. No sé si en sorgo igual sí que hay algo, que como que también está adaptado a la sequía, es posible que se haya hecho más trabajo.
Muchas gracias.
Hola.
Y tu Genome Wide Analysis. Me gustaría saber cuál es tu opinión. Genome Wide Analysis.
Ajá. Ajá.
Entonces haces el ejercicio filogenético
Es una pesadilla.
y tienes una pesadilla.
Sí.
Y estamos un poco como en este intercambio de ideas y nos gustaría saber cuál es tu experiencia en los Genome Wide y con asignación de nombre de genes.
Sí, no, es una pesadilla. No tengo la solución, me encantaría. Sí, nosotros estamos intentando hacer esto porque, por ejemplo, tenemos la anotación en el genoma del pan, ¿no?, del free threshing, este T2T, que es muy buena. Tenemos la anotación de nuestro trigo de referencia salvaje y generamos estos draft genomes de otros 2 trigos salvajes. Y entonces queríamos hacer un lift-off, ¿no?, para ver dónde están estos genes en estos trigos, pero al final incluso el nombre del gen entre el bread wheat y el wild emmer wheat son distintos. Y a la aproximación puede ser hacer un blast, pero después puedes tener gene copies. O sea, al final, hasta donde yo sé, la gente lo que hace es trabajar con herramientas tipo OrthoFinder, que lo que te hace es agrupar los genes por grupos y de modo como jerárquico, y después te puede decir por cada especie cuántas copias, o sea, hay el clúster 1 y te dice cuántas copias hay en cada una de las especies de este gen, y esto es independiente de la notación, está basado en genómica comparativa. Pero sí, ir a gen específico y encontrar este gen así en otro genoma es muy complicado. Bueno, es un rollo, sí, es muy complicado.
Muchas gracias.
¿Alguna otra pregunta? En Zoom ya no tenemos. No. Bueno, pues entonces muchas gracias, Laura.
Muchísimas gracias a vosotros.